Have you ever wondered how your body takes that extra piece of bread or that sugary drink and turns it into long-term energy storage? Yeah, we're talking about fat. Today, we're going to pull back the curtain on the incredible biochemical factory that makes this happen, a process called de novo fatty acid synthesis. So, here's our game plan. We're going to break it all down into 5 simple stages. First, we'll talk about why this process even starts. Then, we'll get into the logistics, the citrate shuttle. After that, we'll look at the molecular assembly line itself, how it's controlled, and finally, why any of this actually matters for our health. Okay, so let's start at square one. Why does our body even go through the trouble of making fat from scratch? Well, it really all boils down to energy balance. This is the big question, right? It's something we all experience, but the biochemistry behind it is just fascinating. The answer is hidden in a pathway that our bodies evolved to make sure not a single calorie goes to waste when food is easy to come by. Now, the official term for this is de novo fatty acid synthesis. De novo is just a fancy way of saying from new or from scratch. It's an anabolic pathway, which means it's all about building things up. It's the exact opposite of breaking fat down. It only really kicks into gear when our energy tanks are already full from things like carbs and proteins. So for this to happen, the conditions have to be just right. We need plenty of extra glucose and ATP. The main headquarters for this operation, that's the liver. But it also happens in other important places like our fat tissue and in lactating mammary glands. And here's a really key detail. All this synthesis happens out in the cell's main workspace, the cytosol. Meanwhile, fat breakdown happens inside the mitochondria. The cell is smart. It keeps these two opposing processes in separate rooms. All right, our factory is officially open for business. So what's next? Logistics. We've got to gather all the raw materials and get them to the factory floor. The cytosol. To build a fatty acid from scratch, you're going to need 4 key ingredients. First up is Acetyl-CoA. That's our main 2-carbon building block. Then you need NADPH, which provides what we call reducing power. Think of it as the electrons needed for construction. Of course, you need ATP for energy. And last but not least, you need a little bit of CO2 for a critical activation step. Now, for any of you studying for exams, here's a great little mnemonic to remember those 4 things. Just think a cat needs power. That's Acetyl-CoA, CO2, ATP, and NADPH. Super handy way to keep those straight. Okay, but we've hit our first snag. Our main ingredient, acetyl-CoA, is made inside the mitochondria. And the inner membrane of the mitochondria might as well be a brick wall. It absolutely will not let acetyl-CoA pass through. So you see the problem, right? We've got our building blocks trapped in one part of the cell, but the factory is in a completely different area. How on earth does the cell solve this? Well, it uses a very clever molecular taxi service. And that solution is called the citrate shuttle. It's a beautiful 3-step process. First, inside the mitochondria, acetyl-CoA hops onto another molecule called oxaloacetate to form citrate. Now, citrate has an all-access pass. It can waltz right out of the mitochondria into the cytosol. Then, an enzyme called ATP citrate lyase acts like a pair of scissors, snipping the citrate back into 2 pieces, releasing our acetyl-CoA right where we need it. Problem solved. Okay, our raw materials have been delivered to the factory floor. Now it's time for the main event. Let's see how this molecular assembly line actually works. It's time to build. The very first step on the assembly line is the most important one. It's the committed step, the point of no return. Our acetyl-CoA gets converted into something called malonyl-CoA. The enzyme in charge here is the star of the show, acetyl-CoA carboxylase, or just ACC. This step uses up some ATP and needs a little help from biotin, also known as vitamin B7. This single step is the main control knob for the entire pathway. So who does the actual building? It's this massive, incredible piece of machinery called the Fatty Acid Synthase complex, or FAS for short. You can think of it as a giant multi-tool with 7 different enzyme functions all built into one single unit working together in perfect harmony. Now, this FAS machine runs a simple 4-step cycle over and over again. First, condensation, where it adds 2 carbons to the growing chain. Then, a reduction, where it uses that NADPH we talked about. That's followed by a dehydration, where a water molecule gets kicked out. And it all wraps up with a second reduction, again using NADPH. So that's the rhythm: condensation, reduction, dehydration, reduction. And it just keeps doing that. This four-step cycle repeats 7 times. Each time through, it adds another 2-carbon unit. After those 7 cycles are complete, the machine is done. It releases its final product, a 16-carbon fatty acid called palmitate. That is the main thing this whole assembly line is designed to make. And get this. Think about the power required. To build just one molecule of palmitate, the cell has to spend a whopping 14 molecules of NADPH. It just goes to show you how energy-intensive building things up from scratch really is. Now, a factory this powerful and this energy-hungry can't just be left running all the time. So how does the body regulate it? This is all about controlling the when of synthesis. The regulation all comes back to that key enzyme, ACC. Think of it like a control panel with on and off switches. On the on side, you've got things like citrate, which tells the cell, hey, we've got plenty of building blocks. Insulin also hits the on switch, signaling that we're in a fed state. On the off side, the final product, malonyl-CoA, acts as a feedback signal to shut down the line. And the hormones that signal a fasting state, like glucagon and epinephrine, they slam on the brakes too. So we've made palmitate, but the story's not quite over. We still need to talk about the finishing touches, and more importantly, connect all this biochemistry to why it matters in a clinical setting. You see, palmitate is just the basic model off the assembly line. The cell can customize it. It can be elongated to make it longer, or desaturated, which just means adding some double bonds to make unsaturated fats. But, and this is a huge but, our enzymes have a limitation. They can't add double bonds past the 9th carbon atom. This is exactly why some fatty acids, like linoleic and linolenic acid, are essential. We literally cannot make them. We have to get them from our food. This little table here is an absolute goldmine for review. It puts synthesis right next to its polar opposite, beta oxidation or fat breakdown. Just look at the differences. Lactation, cytosol versus mitochondria, coenzymes, NADPH for building versus NAD+ and FAD for breaking down, and the big one, hormonal control. Insulin fires up synthesis while glucagon fires up breakdown. Really understanding these contrasts is key. So what happens when this perfectly regulated system goes haywire? Well, if the factory is working overtime, it can lead to things like fatty liver disease. It's also a major contributor to obesity and metabolic syndrome. And on the other hand, if you're not getting those essential fatty acids we talked about, it can cause real problems like skin issues and poor growth. And that brings us to a final thought. This system for storing every last drop of energy was an incredible evolutionary advantage for our ancestors when food was scarce. But now, in our modern world where high-calorie food is everywhere, you have to wonder, has this brilliant survival tool become our biggest metabolic weakness? It's definitely something to think about.